Imagine a future where treating bladder cancer doesn't always mean invasive surgeries or recurring battles with the disease. That future might be closer than we think, thanks to a tiny molecule called miR-21. Bladder cancer, one of the top 10 most common cancers globally, often requires drastic measures like bladder removal, and even with advancements in treatment, aggressive forms tend to come back. But here's where it gets exciting. Researchers from the Doer Institute for Research and Education, IDOR, have uncovered a potential game-changer. Their study, published in Biochemical Genetics, reveals that blocking miR-21 can halt the growth and spread of bladder cancer cells, offering a glimpse into less invasive, more precise therapies. But what exactly is miR-21 and why is it such a big deal? miR-21 is part of a family called microRNAs, which act like genetic editors. Instead of producing proteins, they decide which parts of our DNA get read or ignored, essentially controlling how our genes behave. When miR-21 becomes overactive, it silences genes that normally act as natural brakes on cell growth, fueling the development of cancers like brain, liver, ovarian, breast, prostate, and bladder tumors. This makes it a key player in cancer's unchecked proliferation. And this is the part most people miss. The study focused on a specific gene called REC, which is one of those crucial brakes. When miR-21 is overactive, it suppresses REC, allowing cancer cells to grow wildly and invade surrounding tissues. By inhibiting miR-21 in lab-grown bladder cancer cells, researchers found that REC levels increased, while MMP9, an enzyme linked to tissue destruction and tumor spread, decreased. The result? Cancer cells lost their ability to migrate and form colonies, significantly slowing tumor growth. The findings didn't stop in the lab. By analyzing patient data from the Cancer miRNome database, researchers confirmed that miR-21 levels are significantly higher in bladder tumors compared to healthy tissues. This not only makes miR-21 a promising target for therapy, but also a potential diagnostic tool, a double win for patients. But here's where it gets controversial. While the results are promising, the experiments were conducted in vitro, using high-grade bladder cancer cells. Animal studies and clinical trials are still needed to confirm these findings. Some might argue that the leap from lab to real-world treatment is too optimistic. What do you think? Is this a breakthrough worth celebrating, or is it too early to get our hopes up? Regardless, this research lays a solid foundation for new therapeutic strategies, especially for aggressive cases that currently rely on high-risk surgeries. For patients facing a bladder cancer diagnosis, this could mean more effective, better-tolerated treatments on the horizon. The journey is far from over, but the potential is undeniable. What excites you most about this discovery? Let's discuss in the comments.